Yes.
Yes.
So I have the great pleasure of introducing Professor Kunihiko Kaneko as today's BIMAC colloquium speaker. Dr. Kaneko has investigated fundamental and universal principles of complex biological system by using physical and mathematical approaches. And Dr. Kaneko was a director of the Universal Biology Institute and Center for Complex Systems Biology in Tokyo University. But as you can see in his title slide, he just retired from the Tokyo University 1 week ago. But surprisingly, he will begin new position at Niels Bohr Institute in Denmark in a month. Yeah, I was very surprised when I heard about it. Yeah, I think he still wants to study more. Yeah, so,
I'm not sure.
Yeah. And his most recent research work and the topic of his talk today is universal biology in adaptation and evolution, dimensional reduction and fluctuation-response relationship. So please join me in welcoming Dr. Kaneko.
Okay, so yeah, 감사합니다. So 안녕하세요. 저는 카네코 쿠니히코 스입니다. Okay, so that's all. My Korean.
Wow.
Okay.
Your Korean is better than our.
Okay, so today I talk about this kind of topic, and so universal biology. So my standpoint of universal biology is that, okay, life system usually consists of diverse components. So for example, in a cell, there are thousands of molecules. But as a cell, as a whole, they keep some kind of state. And so as a unit, they are sustained and reproduce as a whole. So microscopically, there are diverse components, so very, very high dimensional, thousands of chemical species, for example. And then macroscopically, maybe there is some kind of control as a whole. So maybe low dimensional description. So this kind of structure can be seen molecule, cell, and cell tissue, and many other levels. So the guiding principle here is there must be some kind of macroscopic, microscopic consistency so that the cell state is stable and sustain itself and steadily grow. So from that constraint of this consistency, we may see some kind of universal law there. So that's my standpoint for universal biology. And with this standpoint, we have been working on some kind of cell reproduction versus molecular replication, adaptation, cell adaptation, and a higher level differentiation, cell and multicellularity, and also kind of different time scale, genetic and phenotypic change. So slower evolution and faster adaptive changes. And so basically, so molecule, cell, and cell multicellular, and sometimes ecosystem. And the point is that this kind of relationship is mutual. So molecule produces cell, but within this cell, so molecule reaction process is so governed by the enzyme concentration and some others. And in the multicellular organism, so each cell state is differentiated according to this multicellular environment. So this relationship is mutual. But they keep some kind of consistency. And from that consistency, it's some kind of steady, stable state is generated. And from that, I hope there may be some kind of general laws therein. And that's also true for evolution, this genotype, phenotype. So this changes slowly and this changes fastly, but there may be some kind of a relationship. So today, I mostly focus on this level, so not this multicellular we have worked on this, but today I'm afraid I don't have time for this. So consistency, so this molecule and cellular level. And from that, I show that some kind of dimensional reduction of these high dimensional phenotypic states through adaptation and evolution. So that's a response theory. And for the second part, I talk about fluctuation that kind of leads to some kind of variance or fluctuation due to noise is correlated the variance due to genetic change. So that's a quite different time scale phenomena, but there may be some kind of correlation. So basic setup today is that, so I consider a cell, and in a cell, there are abundances of many, many components. So for example, in a bacteria, there are about 4,000 or 5,000 protein species or messenger RNA species. So if we consider the abundances or concentration of each component, basically 4,000, 5,000 dimension. So very, very high dimension. So that's a phenotype dynamics. And genotype is a DNA sequence, and that governs the rule for dynamics. So this produces some enzymes, and according to these enzymes, these reaction dynamics are so governed. So this is a rule for dynamical systems. And according to that, phenotype changes. And there are some fluctuation and other. So we are interested in this kind of genotype-phenotype mapping. And according to some genotype-phenotype mapping, there may be some kind of general laws, I hope. So I talk a little bit about the experiment, and this is basically based on transcription analysis of bacteria E. coli. And as for model, so high dimensional dynamical systems model, I mainly talk about this catalytic reaction network model for this cell growth. And the theory is some kind of low dimensional constraint in high dimensional state. So also, please interact me anytime when so you have some questions. So please do not hesitate. And okay, so I start from a very, very trivial law in adaptation. So if we focus on steady growth of cell, so there should be some kind of universal constraint. So that's a very simple thing. So if you have maybe thousands of components, all components have to be roughly doubled before cell division for a cell to produce the almost same cell type. So if we consider that each component abundance Ni grows with some ratio, mu I, this growth rate for each component. So this grows exponential mu I. But mu I should be equal across all components. Otherwise, some components grows much, much larger and some components are diluted. So then this concentration after cell division becomes totally different. So roughly speaking, all mu I should be equal. So if you have m components, mu1 equals mu2 equals mu m leads to m-1 constraints. So if we consider m-dimensional phenotypic space and slowly change the constant environmental condition, then so something like a quasi-static change in thermodynamics, if you change a little bit and change a little bit, then this change follows one-dimensional line because there are m-1 constraints. So that's a very trivial idea. And so we can formulate this very easily. So instead of considering these abundances Ni, consider concentration Xi, so divided by volume. And then the temporal change of this concentration Xi, so X1 to X1000 or something. So it's represented by the change of this. So that is basically synthesis or dilution, degradation, and includes all many, and so depending on other components. So it can be any function of this. And then with this cell growth, So this growth rate mu. So every component concentration is diluted. So minus mu xi. So the steady growth condition is that this equals 0. So that is given by this. So instead of taking xi itself, sometimes it's convenient to use log x and using this fix xi, this, and then we can rewrite this. So the steady condition is given by this. Here fi and E is some kind of environmental condition, for example, stress condition. And so assuming some parameter, so governed by strength of this stress, then this is given by E, so some parameterized. And so this condition is given by this. So far, very, very much trivial. What we assumed is that steady growth, and maybe xi is the concentration. And so this concentration is not 0 because we take log. So that we assumed. And then instead of taking this and then slightly perturb E and assuming that the change is not so large and linearize everything from this equation, then this equation, so Fi, and then the change is given by Jacobi matrix, and mu also changes. And according to the change E, this Fi function also changes. So basically, Jij delta x. So this is due to the change of this Fx. And gamma I delta E is due to the change E. This Fi function changes. So it's some kind of susceptibility or something like that. And then the growth rate change delta mu. So that's just linearization. And then from this, so by taking the inverse matrix of J, we get this. And so this is basically what we are doing, that this given some environmental condition for some type of stress and increasing the strength of E. So with this, for example, this value and along this, and of course, this may be nonlinear, but assuming that linearization. And if we take this and take a different strength of E and E prime, and E and E prime, and take this, and then this part is common because this is computed around this origin. And so we get this for across the different E and E prime, and this follows this equation. The point here is that this side involves the component J, each component, 1, 2000 or something like that. But this right-hand side does not involve J because mu is the growth rate of a cell as a total. So that means if we put delta xj for given E and delta xj for another strength of E prime, all points change linearly. So this slope is same. So we can check this experimentally. So actually, this is the experiment by this Yomo's group. And what they did is that put E. coli into a different strength of stresses. So for example, put some heat, increase the temperature, and a little bit, and a higher increase. And so this axis is this E prime and E in this equation. And what the theory says that all change across J should follow the same line. And here, each point is this different messenger RNA species. So they measure the messenger RNA concentration. So actually 4,000 messenger RNA concentration. And okay, so here are 4,000 points. So it seems to be okay. And furthermore, this slope, according to the theory, this is given by the growth rate change, ratio of the growth rate. And they can measure separately from the experiment. So put this E. coli, and if you put some heat, the growth rate decreases. Actually, this decreases from the original to here, and higher stress decreases more. And then we can measure delta mu separately. And actually, this is the slope. So it looks like okay. And this is a starvation stress. And this is osmotic pressure. And osmotic pressure, there are some deviation for some messenger RNA points. But here, 4,000 points. So the deviated points are just 10 or something. So for most parts, this agrees rather well. So one interesting point here, this theory works somewhat too quite well because we assumed linearization. And usually in physics, when we use linearization, the change should be small. But actually, in this experiment, sometimes they put a very strong stress. And the growth rate from the original bacteria to this later bacteria, stress bacteria, is that 20% from the original. So very, in some sense, very strong stress. If they try to put higher stress, maybe the steady growth of cell cannot be so assured. So they stop that experiment at that stress.
I see.
So up to that range, this seems to be okay.
Right.
So that's one mystery. And another mystery here is that so far they compared with the same type of stress. And that's the reason, because for same type of stress, we go this. But across a different type of stress, may they go to a different line. And actually in the theory, we use this gamma I. And gamma I is that so susceptibility for different type of stress, and this gamma I changes a different type of stress. So gamma I changes depending on the stress type. So we cannot divide this. So then it depends on J. So the theory cannot say anything about across a different type of stresses. But of course, in experiment, we can check that. And this is, for example, osmotic pressure versus starvation, and heat versus starvation. Maybe, of course, compared with the previous one, of course, this linearity is much more blurred. But still, there is common trend and highly correlated. Maybe somehow heat versus osmotic pressure, there are more deviating points. But still, again, here are 4,000 points. So mostly along this line.
Could I interrupt you a little bit?
Yes.
Could you re-explain how different type of stress show up in the equation?
Okay. So this, with this linearization approximation, and gamma I is that the change of this Fi function against the stress type E. And so it's a kind of susceptibility in terms of physics. So, but delta E, in a different type of stress, this depends on the stress type, gamma I. So this gamma I is a function of stress type, some stress type. So one type of gamma I for given stress type this, and for different type of stress, gamma is this. And so previously I said that, okay, compare with the different E and different E prime, and okay, this is common, so and divided, and we get this. But actually gamma I for different stress type is different. So this remains. So we cannot simply derive this.
I see. Thank you.
Yeah. But somehow, okay, maybe not so bad. So basically, okay, this can be also checked instead of messenger RNA. So according to some data, we checked this protein change. So again, this change. So across different conditions, this follows. And again, this slope, this and the growth rate change follows roughly same across many different type of stress conditions. somehow the protein data works better than messenger RNA data. We do not know why, but anyway. So the non-trivial point here is kind of emergent deep linearity. So first, large linear regime and validity across different environmental conditions. So this cannot be explained just steady growth linearization model or theory. So maybe there are some missing points here from the experiment.
Right.
And actually, bacteria is not just a steady growth system. It's a kind of evolved system under many different environmental conditions and many different perturbations. So maybe this is a property after evolution. So, but we do not know what type of bacteria before evolution. So we already have this bacteria after evolution. So we cannot check this experimentally. So we take a very simple toy cell model with many, many components and evolve this toy cell model under some condition and check if this property emerges after evolution or not. So of course, we do not know a very good realistic cell model because it's too complicated. So we take a very simple toy cell model, and that's we discussed about 20 years ago. And so very, very simple abstract toy cell model. There are several chemical species, and all chemical species are catalytic component. And so, for example, some catalytic molecule is produced from other catalytic component and catalyzed by some other catalyst. So XK is produced from XI, catalyzed by some other XJ. And so basically this is the model. And so this is just catalytic model, but catalyst. But we assume there are some resource chemicals. And actually in this model, we have 10 resource chemicals. So basically, resource chemicals are transported from within to cell, catalyzed by some transporter chemical. So for example, externally, there are some X0 nutrient, and X0 is transported, catalyzed by this transporter X2 in this example. And then this is transformed to some other catalyst and catalyst due to this catalyst reaction network. And assume that if the total number of molecules is larger than some threshold, then we divide this cell into two and repeat the simulation. And we take a stochastic model. So for example, we have maybe 100,000 chemical molecules here, and within each, maybe here 1,000 chemical species. So X1 to X,000. And the genotype here is that reaction. So genotype determines the reaction network. So this X1 to X5. And initially, this is chosen just randomly. And so that gives this. But according to the change in genotype, mutation, this reaction network slightly modifies. So instead of X1 to X5 and maybe some other new catalyst appears and X3 produce X1 to X10 or something like that. So slight modification of the reaction network. And the fitness here is the growth rate. So if this reaction works well, and then this is changed to some other and catalyzed by some other and transporter is produced and this can transport external nutrients well. And then depending on this nutrient condition, they can grow well or not, depending on the network. So by mutating the network, then we select those cells that have a higher growth rate. So that's a simple toy model of evolution. Okay, so we put some kind of initially 10, so we put 10 nutrient components, E1, X8, and to X10. And so this environmental condition, all nutrient concentration is same in this simulation, initial simulation. And put this environmental condition and change the network a little bit. And then, so repeat this evolution process. And then according to evolution, then the growth rate increases. So this is this before evolution, and this is after evolution. And then, so to compare with this experiment, we put the stress to this evolved cell or initial cell. This environmental stress is that change this concentration. So for example, we put some kind of vector to E1 to E10 and perturb this. So this is a new environment. So this, during the evolution, this cell is not experienced. And then, so what happens? Also, if how this cell adapts or responds to this new environment.
Mm-hmm.
And according to this experiment, what they did that we compute the change of each concentration of each chemical, delta X. So actually log concentration of each component. So delta X1 to delta X1000. And then for given environmental condition and another so given stress condition. So one type of stress, another type of stress. So basically that's quite similar to this experimental situation. And so actually the point here, this blue point here, is that before evolution, it's just random network. And after 10 generation of evolution, and this is after 150 generation. So this red one. And each point is a different chemical component. So after evolution, there is some kind of linearity across the different environmental stresses. And we computed this correlation coefficient across many, many different environmental conditions, environmental stresses. And after evolution, this correlation is close to 0.9 or something like that for most network and across many different component environmental conditions.
I see.
So maybe so this kind of deep linearity evolves after evolution.
I see.
So this confirms this experimental observation. But from the simulation, we can discuss why this kind of correlation appears.
Right.
But here there is 1,000 chemical species. So the change is 1,000-dimensional space. So it's very difficult to see 1,000-dimensional state space. So we take principal component. And so here principal component 1, 2, 3. And what we plot here is that across many different environmental stress. So each point corresponds to a different environmental stress. And so each point here is that originally 1,000-dimensional space, but projected to this 3-dimensional principal component space. So you can see across many different environmental stresses, they follow roughly one-dimensional manifold. So many, many different environmental change, this follows roughly one-dimensional manifold. But before evolution, there is no such structure. So after evolution, the environmental response is constrained onto a low-dimensional or one-dimensional manifold. So we can discuss why this kind of structure appears. So still, this is a kind of, before that. So change. So this is gray plot is that response against across many different environmental conditions. And actually here, red point is that due to genetic change. So for given environmental condition, and we put some kind of mutation. And somehow mutational change also follows the same curve. And this is the change due to noise. And this follows also the same manifold. So every change due to environmental condition, due to mutation, due to noise, follow the single kind of manifold, major dominant mode. So the argument for this is that, so after evolution, the cell finds a kind of high fitted state. And such kind of state may be quite rare in this high dimensional phenotypic space. And also, this should be robust to many perturbations, because in the simulation, we take a stochastic model, just so. So that means very, very strong noise. So after noise, this should come back to this original fitted state. Otherwise, it can go to a non-fitted state. So fitted state should be rare. So there may be strong attraction to a good fitted state. So this grows strong attraction. But this state should have evolved or still evolving. Along this evolutionary change, there needs to some change. Otherwise, this cell cannot evolve. If the state is completely the same, there is no more evolution. So along the direction of evolutionary change, this can be changeable. So that means for most change, this is attracted to grow strongly. But for the evolutionary change, this follows more plastic. So that seems to be consistent with this two-dimensional manifold, and this is along to this evolutionary change. This is due to genetic change. So this argument seems to be consistent with this observation. And if we assume this structure, so consider this eigenmode, this along this W0, and then that means with this Jij of this Jacobi matrix, one mode, one eigenvalue is very close to 0, and others are largely negative. So instead of taking this Jij, basically we can project everything to this 0 mode, so that is close to 0 exponent. Then, so we can simplify this kind of equation by taking only this W0, this direction of this eigenmode. And then we can simply calculate, maybe you can see that everything is just projected to W0, so we can get this kind of thing rather easily. So basically, the change is along this W0. And then every change due to many different environmental conditions is proportional across many, or almost all components. So far, okay. And also we can check that actually this eigenvalue, one eigenvalue becomes close to 0. And here we computed this eigenvalue of Jij, and this is inverse of this eigenvalue. And so one eigenvalue goes to close to 0. So this is inverse of this eigenvalue. And while others are so quite separation. And actually eigenmode, so eigenvector for this is, so this direction of this eigenvector and principal component is close to, okay, this is 1.0. So it's aligned. And actually this direction is also proportional to the change of the fitness. So along this. So far, okay, this is okay. But then recall that phenotypic change due to environmental variation, mutation, and noise are constrained along this eigenmode. So previously, I discussed this delta x E versus E prime in a different environmental condition. But we can discuss E versus genetic change, mutation. And this should follow almost the same kind of linear relationship. So instead of delta x E versus delta x E prime, we can consider delta x E versus delta x genetic change. So environmental change, E, and genetic change. And then again, according to this argument, this is proportional to the growth rate change. And this is so across all component I. And one interesting point here is that consider some kind of stress condition, E, environmental E. And then try to evolve this cell or bacteria under this condition. Then delta X changes, so transcriptome changes, and the growth rate also changed. And usually with this evolution, this growth rate is recovered. So initially delta mu is largely negative, but this is reduced. So delta mu G should be smaller than delta mu E, this absolute value. So that means, so through the evolution, this slope should be less than 1. So initially many component changes, but then this change is reduced across this evolution. And so we can check this experimentally. And actually, this is the experiment by Furusawa's group. What they did, again, E. coli. And originally, E. coli growth rate is somewhere here. And they put this E. coli into some stress condition. And actually, they put ethanol. And then the growth rate from here to here decreased with this environmental change. So it's somewhat a large change. And then put this E. coli under this ethanol condition and evolve this. And through this evolution over many, this is almost 1,000 generations. And then the growth rate is recovered slowly, not perfectly, but the growth rate is. And then to compare with this theory, they measured this kind of transcriptome analysis, messenger RNA concentration at some generation, and compare, for example, at this generation, and this transcriptome change from this original to here. So that is delta X G. And then compare with this delta X E. So before evolution, this only evolution, environmental change E. So this axis delta X E, and this axis delta X G. And so again, here is this each messenger RNA point. So there are 4,000 points or something like that. And actually, the points are much more scattered if we compared with these environmental stresses. But still, recall that there are 4,000 points. And so most points are around this. And so we can compute the slope here.
Here is a question from in the chatting room. So could you explain what the fitness constraints were during the simulated?
Oh, in this experiment? In this experiment, basically, they select a higher growth cell. So by mutation, some grow faster and some do not grow so fast. And so basically, if they choose randomly from this mutated cells, higher growth cells are selected. So the fitness is just the growth rate of the bacteria. Is it okay?
Yeah.
Yeah. And so then, we can compare this slope and this growth rate change, delta mu E versus delta mu G. So basically, growth rate changes from delta mu E here. Delta mu G is here, or maybe later generation here, later generation here. So this is delta mu G divided by delta mu E. So initially, this is quite large, and then this decreases because delta mu G becomes smaller. And now here, each point corresponds to a different generation. Generation here, 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 here. And for each generation, we computed this kind of messenger RNA change. And the slope is measured. And the slope is this. And this growth rate change is here. So the theory says that these two should agree. So basically diagonal line. Okay, so across over generations, I think this is okay, rather fits rather well. So this theory that, okay, this theory seems to be okay due to this experiment. And another interesting point here is that they repeat this evolution experiment. So for maybe several 8 strains or something like that. And so this is different curve. And actually they computed the principal component across many these so messenger RNA transcriptome spaces. And so this is PC1, PC2, PC3. And actually there are 7 strains. So 7 repeated experiments. And across the different evolution experiments, they follow basically the same evolutionary curve. Interesting point here is that the mutation occurs in a different point for these experiments. So mutation sites are different by strains, but common traits in phenotypic space, the same phenotypic space change occurs. So replaying the tape of evolution, maybe genetic change that is randomly mutating, that can be different. But phenotypic path is constrained along the same curve. And this relationship is also observed independent of this each strain. And we can check this numerically also. What we did is that the previous model, and then here we make a kind of different environmental condition, different nutrient patterns. And then the growth rate decreases. And then we repeat the mutational evolutionary change. And then the higher growth rate cells are selected. Again, fitness is just the growth rate. And we compute some generation here, some generation here, some generation here, some generation here, and the change of this delta X. And so for delta XE versus delta XG for some generation, and delta XE, delta XG later generation, and delta XE much later generation. So basically the slope decreases, and then this slope versus this growth rate change follow this same diagonal line. So that seems to be okay. And also this, we can check this, why this kind of common trend or across many different strains occurs. We did that, repeat this experiment again, evolution experiment, and strain test 10. And again, in this PCA space, they follow the same curve. And actually, they followed basically the original dominant mode of this slow manifold. Evolution occurs along this slow manifold, dominant mode. So that gives some kind of common trend. So far the message here is that, so even though phenotypes changes are high dimensional, but their adaptive change are drastically restricted into a low dimensional space. And this is a result of steady growth and evolutionary robustness. And phenotypic evolution is rather deterministic, even though genetic changes are stochastic. So far we discussed this kind of response by evolution and response by environmental change across many components. But according to the statistical physics, fluctuation and the responses are 2 sides of the same coin. So because due to the Einstein's Brownian motion theory. And actually previously, okay, a long time ago, we discussed the evolutionary fluctuation response relationship. That is response by evolution and just variation due to noise, fluctuation by noise are proportional. And actually this is a little bit old experiment what we did is that evolve bacteria to increase the fluorescence in some protein in this cell. So we put some kind of protein, fluorescent protein into this bacteria. And we did some kind of mutation selection experiments to increase the fluorescence. So initially low fluorescence. And then, so by making some mutants, so this is higher fluorescent one, and this is selected. And then the second generation, this is selected. And the third generation, this is selected. So this is repeated. So through these 10 generations, so this fluorescence increases. And what we plot here is the distribution for isogenic cells, clone cells. So the same gene bacteria. So no genetic change here. And so we have fluctuation. So now we know that even for same gene bacteria, there are large fluctuation for protein abundances and, for example, protein fluorescence here. And you can see that this variance decreases. So after evolution. So basically, evolutionary step here seems to decrease. And also variance decreases. And here, evolution speed is plotted according here. So basically, the increment of this fluorescence per generation, that this axis. And variance here is this just variance for the same gene bacteria. And this variance versus evolution speed, so this red point here, so forget about the black points. And this seems to be highly correlated. And we did the same thing, a similar thing for this cell model, previous cell model. And again, fluctuation and increase in the fitness, also proportional. And then we found that there is a famous theorem by Fisher. Fisher says that variance due to genetic change is proportional to evolution speed. That's the famous Fisher theorem. In this case, so you can consider different bacteria with a different gene. And for each gene, phenotype is different. For example, fluorescence is different. And the variance of this is computed for this phenotype. So initially, genotype is fluctuated, variance distributed. And this genotypic variance is mapped to phenotypic change. And this variance due to genetic change is proportional to evolution speed. That's according to Fisher. This is somewhat reasonable because what they select, then the good, so for example, good phenotype is selected if you select this. And if this variance is larger, maybe this step for evolution is larger because this change due to genetic change is heritable. But in our case, this is isogenic phenotype variance. So it's the same gene bacteria, but due to noise, this is fluctuating. So the next generation of this may become this. Next generation becomes this. So this is just due to noise. But what we have observed, this variance due to noise is also proportional to evolution speed. So that means, so this variance due to noise and variance due to genetic change should be proportional according if these two are true. And actually we have confirmed this, for example, in numerically, this variance due to noise and variance due to genetic change are proportional across through the evolutionary course. And so I don't have time to discuss details of this, why this occurs, but the basic argument here is that variance decreases if the robustness increases. So if some state is good and does not change so much, so that is more robust. And if this robustness increases, then the variance decreases. So if the robustness to noise increases, then the variance due to noise decreases. But if the robustness to noise increases, robustness to mutation also increases, because perturbation by noise and perturbation by mutation may be correlated. And then this also decreases. And so this decreases and this also decreases. That's the explanation. And actually, recall the previous result, the variance due to noise here. Okay. Okay, the variance due to noise here, this red one. This is the change by noise. But this is the change by mutation. And this is due to environmental change. So this follows the same curve. So that means variance due to noise and variance due to genetic change follows the same kind of manifold. So we can discuss, instead of the previous study of this response, we can discuss the variance due to noise is also mapped to along this, so one dominant mode. So originally very, very much high dimensional, but this is projected along this dominant change. Then we can discuss that basically this is due to noise, and this is again, so following the same argument of the response, we can show that this variance due to noise and variance due to genetic change is also basically independent of each component and proportional. So for example, we checked this. So in this previous cell model, and each component fluctuation, we measured each component fluctuation due to noise. And then we apply some kind of mutation to each this cell model, and we can compute the variance due to this mutation, genetic change. And so here, each point is a different component. And so VIP due to noise, variance due to noise, and variance due to genetic change across all components, they are proportional. And these are the results for different strengths of mutation rates. So far, this is only for simulation. We have not had experience yet. But anyway, yes.
I have a question, sorry. Yeah. The component means the species in the system?
Yeah, chemical species. So each component of, not the cell species, each chemical component. So in this cell model, yeah, in this simulation, 1,000 chemical components, 1,000 chemical species. Yes, in the previous slide, in this case is a kind of across an evolutionary course.
Okay.
Yeah, so it's in some sense different. Yeah. In some sense, this is much stronger. Yeah.
So what does the average of X mean in the previous slide?
Here.
Yeah, here.
Okay. Here, in this case, only variance of the fitness. Variance of the fitness due to noise and variance due to the genetic change. Only for the fitness. In this slide. And in this case, so all phenotypic space, so all high dimensional space. So of course, this is correlated, so the fitness also correlated, yeah.
Okay, thank you very much.
Yeah, but this is also interesting because VG, according to Fisher, is proportional to this evolution speed. So that means if one component is highly variable by noise, that can be easily evolvable. when you put some mutation, then if this component is, this variance is large, such component is also largely changed by genetic change. So in some sense, we can predict which component can be more evolvable just by looking at the fluctuation before mutation. So that's somehow kind of evolutionary constraint or direction in evolution. So in some sense, we can predict the evolutionary course. So basically, what we have seen that dimensional reduction leads to deep linearity across environmental condition and also fluctuation. So that's the basic story today. And of course, we need further studies to establish the present theory. Maybe we need further confirmation by experiments. And actually, Chikara Furusawa has been working on a bacterial evolution experiments in many different antibiotics. And he has been observed many correlation across the different environmental conditions. And also, so far today, I talked about the confirmation by models by using catalytic reaction network cell model. And we did a little bit on gene regulation network cell model. And again, gene dimensional reduction is observed. And so it seems to be okay. And in a different level of statistical physics model, Sakata worked on a spin glass model. And in this case, so we consider spin glass Hamiltonian, JIJ-SISJ. And then as a fitness, we set up some kind of spin configuration. So some target spins should be aligned, for example, as a fitness. And then evolve JIJ. So this is given by Genotype. And she has found that, again, dimensional reduction is observed. And this seems to be corresponds to replica symmetric state. If you are familiar with spin glass, maybe you know something. And if this real spin glass state, that shows replica symmetry breaking. And in that state, robustness is broken. And this kind of dimensional reduction is broken. But before that, this kind of dimensional reduction is observed. And what she did here, and then also recently last year, Kian-Yuan Tang, who was a postdoc at that time, computed the protein model and protein data, real protein data. And then he found that correlation in this fluctuation by this due to noise or dynamical change along this environmental condition, and also change mutation due to evolutionary change are strongly correlated due to some kind of dynamics and evolution correspondence across many protein residues. So that's another support for this theory. So it's a quite different level, cell model to protein level or spin glass model. So maybe we need some kind of theory for evolutionary dimension reduction, and why, in what cases this dimension along this one-dimensional curve, maybe the real cell, maybe not one-dimensional, maybe a two or three-dimensional. We need to understand why this occurs. So I don't know, we may have some kind of statistical physics theory like the linearization group or collective mode projection. So we need more deeper theory for that. And as for related issues, maybe we don't have time to discuss with this, but maybe, okay, I just touch upon this only. So today I talk about this steady growth state. So the cell can grow exponentially, but in a very bad state. So usually bacteria goes to stationary phase or dormant state. And that state is almost the growth rate is 0. So transition from exponential growth to almost zero growth state. So how this kind of transition occurs, that's an interesting topic. And actually, a few years ago, Yusuke Himeoka proposed a simple model for this transition. And so he could predict. So one interesting point here is that after it goes to a non-growing state, they put some kind of nutrient again. Then after some time, the growth rate is recovered in a good condition. But usually there is lag time. So the cell needs some time to recover the growth. And interestingly, this lag time increases how long it's starved, the time how long it's starved. So if the cell is starved a very long time, you need much longer time for recovery. So in that sense, non-growing state is not unique. They remember how long it's starved. So there may be some kind of good state is single, low dimensional. But bad state may not be just represented one single parameter. And for example, Tolstoy in his novel Anna Karenina, so the first sentence of this Anna Karenina says that happy families are all alike, and every unhappy family is unhappy in its own way. So maybe happy cells are all alike and low dimensional reduction, but every unhappy cells, non-growing cells, may be unhappy in its own way. So maybe dimensional reduction may be broken, but we need some theory for that. So that's still ongoing. Okay, so that's all. So to sum up, so we discussed low dimensional reduction from a high dimensional phenotypic space, and that it may be due to the robustness of this good state. And so from that, we have seen universal law for adaptation and evolutionary relationship and fluctuation law. Okay, thank you very much. So, kansa hamnida. Okay.
Okay, so I think it's question time. So yeah, please feel free to ask a question and unmute by unmuting yourself.
I have a question.
Yes.
So how do you separate the noise by genetic mutation and just noise VIP? How do you separate from the experimental data?
Oh, actually, so for the experiment, so we need to do is that, so in this, for example, in this experiment, for example, here, so for example, this distribution, so they take this one evolved bacteria and then, so produces clones. So 1,000 clones. And then measure the fluorescence. So there is some kind of flow cytometer, and they can measure. So multiplicate these cells. This bacteria can grow very fast. So you can make from this clone bacteria over thousands of prime cells. And then, so these are just due to noise. This variation are just due to noise. So that can be computed. And then this VG, that is somewhat more difficult to compute because if you take a kind of different genes, bacteria, and variance, they include due to both the variance due to the genetic change and due to the noise. So they first need to compute the average, for example, average here across all different noises, and then compute this averaged concentration or averaged fitness across over noise, average against noise, and then make a different mutation, different genotype, the different genotype, and then compute the variance across different mutants. So experimentally, that's not so easy.
I see.
But for numerically, this can be done because we can numerically compute this. So by taking this one type of cell and compute this, put some noise and measure the average for this genetic and make a mutation and then the average and make hundreds of mutants and then variance of this average. So numerically, that's easier.
I see. It sounds like a conditional marginal distribution, I think.
Yes.
Okay.
Yeah.
Thank you.
Yeah.
Thank you so much.
Yeah.
Any other questions, please? Yeah. Uno Song. Yeah, please do it.
Yes. Yeah.
Uno Song.
Yeah.
Please. Yeah.
Maybe muted.
I think he's unmuted. Yeah.
Muted. Yeah. I cannot hear. Yeah.
Can you hear now?
Yes.
Yeah.
Yes.
Thank you for the interesting talk. What in your life they face the multiple dominant single stress, but also the equally can face the multiple comparative stress at simultaneously. So what I'm asking is what happens if the multiple comparative stress? What happened when the multiple stress acting on the simultaneous? Because so there are two cases, only a linear combination, but also there is a property can happen. So I want to hear your speculation for the several variable.
So you mean heat and osmotic pressure simultaneously?
Yeah, simultaneously.
Yeah. so far we have not done. So they have not done this kind of experiment. And actually, recently Chikara has been working on not the simultaneously, but he has been working on the evolution of bacteria across a different type of antibiotics. So there are many different antibiotics. And what he found is that kind of cross resistance. So if this bacteria is strong against one type of stress, one type of antibiotics, this may be sometimes stronger also to a different type of antibiotics.
I see.
But in some cases, if for one antibiotics, if that is strong for evolve to have a tolerance to one antibiotics, then may be weaker against different antibiotics. So then he can compute a kind of a cross correlation for this. So one type of bacteria, and maybe he has now data for maybe 100 types of antibiotics and 100 and kind of matrix. And actually, student of mine is working on a theoretical model to explain that. So what he proposed that maybe he can predict this kind of correlation against a different type of antibiotics before evolution. So I said that before evolution, so just by response or fluctuation before evolution, And this is correlated to this response against evolution. If that is applied to across the different antibiotics, maybe we can predict